morning, everybody. So, this conference is going on since yesterday, and this talk is not directly from physics and from spectroscopy or experimental physics people have been talking about, and this conference is about, but it involves some biological materials. So, it will be like a break, maybe a good break. I'm Naveen from Hazaribagh, Dhanbad University. So I will talk about some forefront, they are computational works mostly, computational works on bio and nanobio materials, which include biomolecules like DNA, RNA, proteins, and I being a student of physics, working on biological systems, so how it is like. It not just involves biology, physics, but also chemistry, mathematics, computation, as we will see along. So Professor Siddhartha Roy was my PhD guy. He has retired. He is still there, but has retired from the institution. He was at Bose Institute, and then later on he became director of Indian Institute of Chemical Biology in Kolkata, Jadavpur. He is a Bhatnagar. And Professor Gautam Basu, he is at Bose Institute again. Maybe he too has by now retired. Professor Surajit Sengupta, he was my co-guide. He is no more in this world. I gave my homage to him a few years back. He was director of TCIS, TIFR Center for Interdisciplinary Researchers in Hyderabad. He was at IACS, Indian Association for Cultivation of Sciences in Jabalpur, Kolkata, and also at SN Bose National Center for Basic Sciences in Salt Lake, Kolkata. And Professor Jaideep Chakrabarti is still at SN Bose. He was my co-guide to begin with, but later on I moved to Professor Surajit Sengupta. And Dr. Soma Samadar, Asit Kumar Manna, and Paromita Raha. They were my colleagues working in laboratory. That was a biotechnology, biochemistry kind of laboratory. I being a physics student, all of them were either chemistry people or biology people. So Soma Samadar is in Lady Prabodne College, Asit Manna is in USA, University of Utah as a research associate probably, and Paromita Raha is in industries. My work overlapped with them and we worked together. So that one is Vinoba Bhave University on upper part and picturesque complex, 130 kilometers from here, and lower one is SN Bose Center in Salt Lake, Kolkata. So the works I will be talking about, the first one, electrostatics and salt effects of protein nucleic acid interactions. By nucleic acid, we might be familiar with them, RNA and DNA, ribonucleic acid and deoxyribonucleic acid. And what is the role of electrostatics, charge-charge interactions, like dipolar water is there in the medium around, and salt ions are there, they are charged again, they are mobile too. So what is the role of electrostatics in protein nucleic acid interaction, protein DNA binding, protein RNA binding, and the computational part? So that rotating structure is DNA, double helical structure of DNA. In 1953 or around, this structure was invented. One physicist was involved in it, otherwise they were biologists, Watson, Crick, and physicist, she was Franklin Rosalind. That was very important at that time. This structure opened a plethora of works in biotechnology and the developments you see today. Because we can go at the molecular level into DNA inside our cells, inside the nucleus, and make changes into genes. That is what biotechnology is about, experimental. And maybe you can change human being to some different species from clones one can prepare. So things are controversial also. It's not just a question of science development, but also of ethics. So this is DNA structure a little bit in detail. Adenine and thymine and cytosine and guanine, they are called monomer units. This is a DNA is a long chain. This little bit of introduction is necessary. So guanine and cytosine, they form hydrogen bond, 3 hydrogen bonds, and adenine and thymine again they form hydrogen and 2 hydrogen bonds. So these two are chains, one chain and another chain, and then bond over each other in double helix form over here. And this is RNA. Again, this also is a chain. It will extend on this side like this, and on that side like that. So this is 5 prime direction, 3 prime end, 5 prime end, 3 prime end. So this is a pentose, which is called a base, a nucleotide, and this phosphate group through which 2 nucleotides are connected. This is one nucleotide, and this is another nucleotide over here, one another here. So they are connected by these phosphate groups. So from 5 prime to 3 prime direction, this is 5 prime, this is 3 prime. So in chemistry, such nomenclatures are used in organic. And these are RNA, DNA. This also is double helical DNA. This is a single helical, small molecules. RNA are relatively smaller. DNA are much longer. Inside our cells, inside the nucleus, chromosomes are there, and those chromosomes are essentially DNA, bound over proteins. So those DNA, if you take out one of it from one chromosome and extend it like this, their diameter of that string will be some 20 angstrom, very small, but the extensor length will be like in say centimeters, even meters, 1 meter or so. So very long string structure, polymer chain, DNA. But RNA are, again, they are also nucleic acids, but the smaller molecules. And here is protein, the third part, the third important molecule, which is amino acids connected by peptide bonds. And again, this becomes a chain. So one amino acid, second amino acid, third amino acid, fourth amino acid on all sides. So that also is a chain. So polymers, DNA, RNA, proteins. So this is a protein chain. This is called peptide bond by which they are connected. You see C alpha, two carbon atoms are there, one nitrogen, again carbon atom, again carbon atom. So one is named C alpha, another is just carbon alpha, carbon, nitrogen. Again carbon, nitrogen, carbon alpha. Again nitrogen, carbon alpha, carbon. This way a backbone, protein backbone is there, and there are side chains, R1, R2, these are small groups. So dihedral angles, these angles have, so this work was done by G. N. Ramchandran, again an Indian scientist, physicist again, working in biology. So this C alpha, C and N positions, atomic positions, they will 3 points, so they will form a plane. And then you take another next from here, starting from here, carbon, nitrogen, carbon alpha. So this will form another plane. So 2 planes, there will be an angle between them, that is omega. And again, go from C and C alpha plane, and then N C alpha C plane. Then 2 planes, angle between them, they are dihedral angles. So that will make phi angle. So omega angle, phi angle, psi angle. 3 dihedral angles. So that fixes the structure of protein. And this again is very important thing in biology. From DNA, which reside inside the nucleus of cell, R cell. So this double helical 2 chains, and from there this process is called transcription. That takes place inside the nucleus by one protein, again RNA structure. So a lot of RNA protein interactions are there in life. They are important. So from DNA to RNA, which occurs inside the nucleus of the cell. This is called transcription. This is essentially mRNA, messenger RNA. From RNA to protein, which takes place, this process is called translation. Translation of protein. Translation of transcription and translation of protein. From DNA to RNA to protein. But this RNA to protein, that takes place, takes place outside the nucleus of cell. I mean, the cell, our cells, they are composed of, the central part is where the DNA and RNA normally stay, that is nucleus, and outside is cytoplasm. So ribosomes and other mitochondrial structure and different many big molecules, they are there in cytoplasm, mostly water and then organelles. So DNA to RNA transcription, that takes place, this reaction takes place inside nucleus of the cell, and this outside nucleus at the ribosome site. So DNA to RNA, this is called central dogma of life.
Sir, you have 3 minutes.
3 minutes. Okay. So more than, I have a lot of results, but I don't think they are important. But they may be important, but for here, because this introduction part is a bit necessary. So from DNA to RNA, this blue one, red to blue, by RNA polymerase, a protein. So, and then that mRNA comes out to ribosome site. Again, the structure of this ribosome, that was again found by one Indian origin scientist, and he got a Nobel Prize for that 10 years back. Venkat Ramakrishnan. He did his graduation in India itself, in Gujarat, but later on worked in London and USA. So he got Nobel Prize in chemistry for this ribosome structure. He got this structure. He is a physicist basically again. So this structure he found using X-ray diffraction experiment. Structure of ribosome. So from DNA to RNA, then RNA to protein. This is protein synthesis. And protein DNA interaction, like here, protein DNA, and here some rRNA. So they are important, protein DNA interactions. So this is electrostatic work. Contour plots, this results. So here a protein structure is there. If you forget this bright red color and bright blue color, then there is a protein molecule, and there is a DNA molecule over here. This is a long chain but cut short, and then we do electrostatic calculations, calculate electrostatic potentials. From there, once you have potential at atomic sites, first you replace the system by a three-dimensional grid. You do finite difference numerical, use numerical techniques and calculate electrostatic potential, thereby electrostatic energy. And once you have energy of the system, protein DNA, you can calculate binding energies. You can separate the DNA protein into 2 parts, and DNA calculation, RNA calculation, electrostatic energy calculation, and then for the complex and difference is binding energy. You can calculate binding energy, association, dissociation constants, and you can compare with results. So because there is not much time, so this is essentially Poisson. This is Poisson-Boltzmann equation. This one is for salt, mobile salt ions. So nonlinear partial differential equation, second order, very difficult to solve analytically. So you use numerical techniques. If you forget this middle term, which is because of mobile salt ions, then this dielectric constant is, if it is not dependent on position, if you take them out, you see this is Nabla square 5. If you take this part to this rho upon epsilon, Poisson's equation essentially, and then you calculate the electrostatic energies, and then binding energies and association dissociation constants. So I will go through them.
Yeah, okay. Sorry, request to conclude.
Okay, so these are the results in comparison with experiments. So this was one work. Conclusions. Molecular dynamics simulation. Just I will take 1 minute. molecular dynamic simulation and dissimulation of a peptide, KYKETDL, part of protein, lysine, tyrosine, lysine, and HCV, hepatitis C viral infection. And our work, molecular dynamic simulation, that was for structure of the peptide, and it was important for stability of beta turn in this peptide for translation inhibitory activity against hepatitis C viral infection and molecular dynamics study. And then the calculation details results. And this one about target search by protein on genome. And this we extended later on. And experimental data we used to SARS-CoV-2 genome, coronavirus, and these results, which are important to be published. Thank you very much. And
thank you, Professor Scotland.
this is the group.
Okay, again, sorry, we cannot allow the interaction at this moment now. So, like, first,